Welcome to the Emerging Technologies Imaging session. My name is Kristen Kwan, and unfortunately one of the things that's not in the program is that I'm moderating this session, but I also have a co-moderator who's with me, my colleague Fabienne Poulin. So today we have 8 really exciting talks up for you guys, and our first speaker is Ben Cox, who's coming to us from Duke University, and he's going to tell us about cell cycle dynamics during scale regeneration.
All right, can I get started whenever? Cool. So thanks, of course, to Kristen and Fabian, and then all of the organizers of the conference, and of course everyone who made it out here in time for the first talk of the session. So I'll be talking about a project that unites a couple of my interests, namely live imaging and computational single-cell tracking, and it unites those interests in the goal of better understanding the main theme of the POS Lab's research, which is, of course, regeneration. Now, first I want to address what we can really understand by taking these techniques and combining them. And this is just a sort of a preview, a teaser image movie featuring osteoblast dividing that I'll get into in greater depth later in the talk. But if we can achieve what I've proposed on this slide, that is capturing all the cell divisions as well as other cell behaviors, like characterizing planes of division, proliferation patterns, and stereotyped patterns of proliferation, then I think we can get at understanding some fundamental concepts about regeneration that we would not otherwise be able to understand if we were limited to a kind of tissue level view of regeneration. So some of these concepts that I think we can better understand would, or these discoveries that would help us better understand regeneration include things like if particular cells or subpopulations of cells in a regenerating tissue contribute more or less through proliferation to the full regenerated tissue, whether there are specific domains of proliferation, whether those domains change over the course of tissue regeneration. And as the title of my talk implied, we use the Zebrafish scale to study regeneration. It's kind of a new direction in the POS lab, but it's a nice model to study specifically osteoblast regeneration because the tissue itself is small enough that it's really easy to image completely, and it also regenerates rather quickly. So thanks to Amy for the artwork. We basically what we just do is take a pair of forceps and denude a patch of skin along the trunk of the fish. Within one day, which isn't shown here, but within one day you just start to see a handful of osteoblasts appear. By 2 days, you really start to get some patterning, and the shape of the scale begins to take place. Begins to appear, and that there are some of the features of the scale begin to appear, and then from about 2 to 5 days, you really see massive growth. After which growth slows down and just really occurs along the edges of the scale. So kind of the take home is that if we want to look at regeneration happening rather quickly, there's a window of just a few days where we see massive growth and can understand. Can get a window into regeneration occurring in a relatively short time scale. So to look at proliferation dynamics, first we want to establish kind of when and where proliferation is happening and what's the contribution of proliferation to regeneration in this particular tissue context, because there are a number of mechanisms that can account for tissue regeneration, such as direct differentiation of a progenitor cell source to osteoblasts, proliferation being another mechanism. So to kind of establish what these relative contributions are, we use a Kaida system under the control of the osteoblast Austerix promoter. It was generated by a former lab member, Samit Singh. So if we just take what we have here, which is a mature adult scale, convert it under UV light all the way to red, and then pluck the scale, we can look a day later when new osteoblasts first start to appear and try to assay what color they are. If we see primarily red osteoblasts, that would imply that there are some left over after plucking the scale, and that the proliferation of those cells is what's responsible primarily for creating the new tissue. Whereas if we see green cells, that would mean they were unconverted and they had come from some other progenitor source that would differentiate into new osteoblasts. And indeed, what we see is just green cells. So we can conclude that the initial source of osteoblasts in scale regeneration is not cells that were left over after plucking a scale or after a scale is lost, but some progenitor source that's differentiating into new osteoblasts. However, if we change these experimental parameters slightly, so not converting the scale before plucking it, but rather plucking a scale off and then waiting until some of those new osteoblasts appear and converting those with exposure to UV light, then looking about 12 hours later, what we find is that the scale, as I implied earlier, has grown massively in a relatively short amount of time. But what we see is that the majority of these cells, in fact over 60%, retain some red label, implying that they were either present or are the descendants of cells, daughters of cells that were present at the time of conversion at 30 hours. And then if we look even a little bit later, converting at around 36 hours, nearly 100% of the cells that we look at after a 12-hour window retain red labeling. So by this point, we're seeing almost entirely growth due to proliferation. So just to sum up this part, we have this initial phase of scale regeneration where a progenitor source directly differentiates into osteoblasts to provide the initial pool of cells that will make up the regenerated tissue. But then later, primarily proliferation drives growth of the scale. And now if we want to study those proliferation dynamics, we can use a Fuchi system. So for those not familiar, the basics of Fuchi is just that degradation sequences of cell cycle regulators are tagged to fluorescent proteins. So CDT1, which is expressed in G1, its degradation sequence is attached to mCherry, and then geminin, which is expressed in SG2 and M phases, tagged to Venus. And here I have, just to get oriented, a juvenile scale developing, so most of the osteoblasts in the scale are in G1 phase, but along the border you see some scales, some cells in the proliferative phases. And I want to, of course, thank Val Tornini, who generated these lines. So in addition to this technology to look at progression through the cell cycle, we need a system to image long-term. So we developed some methods somewhat similar to what was published by the Siegman Lab last year, that allow us to image long-term. So we just combine low-level anesthesia with a reduced concentration of tricaine, circulating water, and partial suspension of the zebrafish under agarose, not suspending the actual tissue that we're looking at regenerating, and not suspending the fish's head so that it can keep gilling and maintain oxygenation. So combining these technologies, we're able to generate movies of regeneration of 1 day or longer, and we're able to image hundreds of cells appearing and dividing over the course of this movie. And by combining cell segmentation software that was previously been written and published by some of the D'Italia Lab's collaborators with tracking scripts that we've generated in-house, we're able to track hundreds of cells and quantify their progression through the cell cycle. So for an example, we have a relatively vanilla osteoblast here, and that it divides. We see geminin signal accumulating after division. That signal disappears as mCherry ZCDT1 accumulates. So we have a pretty typical transition from SG2M after division, accumulation of the G1 protein. And as mentioned, we were able to do this for hundreds of cells. And while this graph may not be particularly helpful, if we dig into some of these individual traces, we can find some really interesting subpopulations of osteoblasts that might help us better understand how regeneration occurs. So what we see in about 6% of these dividing osteoblasts is that they don't actually accumulate any appreciable signal of mCherry's ZCT1. They divide and then immediately begin accumulating the green Fuchi protein again. So we see them go through several rounds of division without entering G1 phase. And again, this happens in about 6% of the cells and might go a ways to explaining how we see such rapid growth occurring in the scale during these early stages of regeneration. To wrap up what we've just discovered so far, we know that there's a progenitor source differentiating into this initial pool of osteoblasts after scale loss, and then after these new osteoblasts appear, proliferation takes over to form the bulk of the regenerated scale. So we've developed some live imaging and computational methods to track cell cycle dynamics in an osteoblast population over long periods of regeneration. And we know that there's a subpopulation of osteoblasts that immediately re-enter the cell cycle after division. And just going forward, in addition to the Fuchi lines that'll allow us to track progression through the cell cycle and look at later phases of regeneration when those proliferation dynamics might change, we've developed other new technologies and transgenic lines to help us characterize even more aspects of regeneration. So this Austerix H2A mCherry line is a more permanent marker, unlike the dynamic Fuchi marker, so we can track through multiple rounds of divisions and as well as look at even more see even more characteristics of these regenerating cells, including plane of division. We can combine those with mutants and pathway reporters to look at things, as I mentioned, like division plane, cell size, membrane dynamics, and the influence of genetic and molecular pathways on regeneration. So that's where we hope to go in the future with this project, keep using our live imaging system and computational tracking to better understand regeneration on the single cell level. So as I mentioned in the talk, I specifically want to thank Val Tornini and Sumit Singh for generating some of these transgenic lines, Amy Dixon for artwork, Alberto Pugliafito is a collaborator of the lab who helped with some of the tracking, script writing, and then of course just my labs. And I'll take any questions with the remaining time I have. Rosa?
Does it form a blastema?
Yeah, Rosa asked if the tissue forms a blastema. I would say, no.
Other regions that would be considered something like that?
I wouldn't think so, because with a blastema, you think about there being like a part of the tissue that's left over that then forms sort of this dedifferentiated mesenchyme and new patterning occurs, whereas this is like you're basically removing the entire tissue, and then there are progenitors kind of beneath the skin. The way that scale development works in the juvenile is that there are signals that are secreted by the basal epidermis that sort of recruit fibroblasts to differentiate into osteoblasts and grow outwards. And so generally we think that the same thing occurs in adult regeneration.
So is there a spatial pattern for the 6%?
We haven't quantified that. She asked if there was like a spatial pattern to the cells that proliferate more quickly, the ones that skip G1. We haven't quantified that definitively yet. They do appear to be more along the outsides of the cell. That would sort of seem to be in agreement with what we see later during regeneration, where proliferation occurs more exclusively along the borders of the scale. So that would seem to be the fastest region of growth along kind of the perimeter of the scale.